word in vitro means in glass. It's not in a living organism. And for example, this is a kind of research you do in a wet laboratory. Maybe you have microorganisms or cell lines or enzymes. And in this case, the research question would be, will you, and remember now earlier we've said plants, yeah? Maybe you have a potential drug from a plant or a potential drug which you synthesized in the laboratory, and you have a research question saying, will this drug, will this a potential chemical will have an anti-cancer effect or antiviral effect. So what we do in an in vitro study is we try to grow the microorganism in a culture media, and then we export that microorganism to that specific chemical, which we are now anticipating it could be a potential drug. And we'll see whether this drug or a potential drug could be inhibiting the growth of the microorganism or the growth of a cancer cell. Also, cancer cells, you can grow them in a culture media. You provide them a favorable environment to grow using a culture media, and then they grow, and then you try to check whether your potential chemical compound, which could be a potential drug, could inhibit the proliferation or growth of the cancer cells. Even, for example, in HIV-related research, you try to see the effect of your drug on viral infected cell lines. So you see, so in all these things, in all this study, there is one thing in common. You're trying to prove a concept where you have a research question saying that, yes, I have a mitishamba, I have a plant extract, but will this plant extract be able to treat cancer or HIV, or will it have antimicrobial effects? So the first thing you can do is you take the sample, you take it to the lab, and now you are not putting it in animals. It's not an animal study. In vitro study, you're trying to see the effect of this extract on a glass, maybe on a growing cell line, maybe it's a cancer cell line, maybe a breast cancer cell line, or it is a herpes simplex virus, maybe it's a human immunodeficiency virus, adenovirus. Then you grow those viruses, and then you try to inoculate your extract to these viral infected cell lines, and you see the virus really still multiplying in presence of your potential drug, or are they dying? So if the cell lines are dying or are not proliferating or are not multiplying, now you can conclude something. Maybe your extract could have a potential anti-cancer effect because it's just killing most of the proliferating cancer cells, or you can conclude saying that maybe your extract will have an antimicrobial effect because it's killing or it's preventing the growth of the microorganisms. Then if you prove this concept, again, the research can go to the next phase, maybe to animal studies. So you see, we don't take drug experiment to humans. First, we need to prove whether this drug works at the lab level. So research done for purpose of drug discovery in wet lab in a glass is called an in vitro study. Other modality of drug discovery is what we call an in vivo drug discovery studies. Earlier video we have talked in vitro, which is a study in a glass. In vivo is a study on a living organism, on experimental animals. We have different types of experimental animals, from small animals to the higher animals, but usually in a research laboratory setup, we start usually with small animals like rodents, like mice, rats. We can also do research on guinea pigs, and also rabbits, and depending on the type of study, the research can go to the higher animals. But why are we really doing a study on animals? You see, the research on experimental animals is based on two important aspects or principles. Number one, we need to ensure safety for humans, because if a drug is going to be toxic to the experimental animal, most likely we can abort the study, because if a drug is causing mortality or signs of toxicity to your lab animals, you may not be interested to take that study to to humans. You see, now that is protecting the human species from toxicity, from dangers of health. The other one is to check effectiveness. You see, you don't need to bring a medicine or a medical product to the market without proving it. But there are a lot of controversies and a lot of pointers with this one. There are some drugs which are really toxic to experimental animals, but very beneficial for humans. Take, for example, some of the anti-cancer drugs, and that is a discussion for another day. But based on the scope of this talk, the in vivo studies which are done on experimental animals should be done in an ethically approved way. Still, because animals have their own rights, so they should be done in a small number of animals in a conducive environment. But any researcher who does research on animals should get an ethical approval to do that research on animals. But remember, these experimental animals are very helpful when it comes to drug discovery. They can tell you, for example, it's very difficult to model or to say a drug could be useful to improve learning and memory. You know, it's very difficult to say a certain compound might cause dementia or can affect learning and memory, but you can try to administer that compound to lab animals and you can try to see whether the learning and memory or cognition of the lab animal is affected. This is a good model which can be tried on. For example, rodents are very exploratory. So we have different types of experimental models to check for cognition. Maybe let me ask this question. What if chewing miraa or smoking weed could have a long-term effect in causing Alzheimer's disease or dementia? It's very difficult to see that on humans because some toxic effects of such drugs could be seen on humans after years of being administration. But you could see these effects in lab animals in a certain protocol, not in a year. You can administer that for 2-3 months and you could see whether such a plant extract, or maybe as I mentioned, maybe Cata edulis or weed could cause dementia or could have some psychiatric symptoms, and potentially it's possible to advise the community at large. Just I took those examples, but you know, there could be many potential chemicals that could be investigated on animals. So animal study has really shaped the drug discovery process. It has really helped to ensure safety of the human species from toxic effect of quite a number of chemicals. Hope you'll have a lot of input, a lot of comments on why drugs are discovered on animals, why rat, why mice. We can have that discussion in the comment section or in another video. See you.